Studying the transport of bacteria in porous systems is important with regards to contamination, spread of disease, and bioremediation. For this, experiments and mathematical models operating at the single cell, the population, and the microbial community level are required. Here we present tools to study bacterial transport using microfluidic devices and microscopy, as well as larger devices in combination with flow cytometry. The combination of microfluidic devices with microscopy and flow cytometry offers a range of possibilities to study bacterial transport phenomena at different spatial scales.
In order to fully exploit this protocol, it is suggested to have previous experience in microscopy, basic image processing, designing microfluidic devices, and mastering flow cytometry.
This video describes two methods to study bacterial transport at different spatial scales. The first method, based on microfluidic devices, relies on microscopy to count individual cells. This system can be used to study bacterial transport across multiple spatial scales, from the pore to the entire porous system scale. The second method, using larger fluidic devices coupled to an automated dispenser and flow cytometry, can be used to study bacterial transport phenomena at the scale of entire porous systems. Such methods can also be deployed in laboratory-scale column studies, as well as small field surveys. To begin, design the desired porous geometry that consists of a matrix of circles. Based on the chosen geometry, prepare a mold using standard SU8 photolithography. Prepare 50 grams of PDMS by adding 90% of elastomer to 10% of curing agent